Hi, this is Dr. Arnold. Can everybody hear me okay?
Great.
Yes.
Okay. All right, Sam, are you ready?
Yes, ma'am.
Okay.
Can you see my screen? It should be sharing.
Yes, I can see it.
Okay, great.
Just not the presentation. Yeah, there we go. So I'm going to go ahead and get started. I'd like to thank everyone for joining us today for Samantha Murphy's dissertation seminar. Sam joined the PhD program in microbiology in fall of 2014 or 2015. I don't remember now.
2015.
Sam started in my lab by looking at whether or not the rotavirus interferon antagonist protein, NSP1, had other ways in which it blocked the interferon response. We really wanted to know if this was a multifunctional protein. And in the process, she learned a lot of new techniques, some of which included the microscopy and subcellular fractionation that she's going to show you today. And in these cases, she started to get some data, which she was showing me that NSP1 protein was localizing to the nucleus of cells. Now, this had really never been observed for any other rotavirus proteins because this virus replicates entirely in the cytoplasm. So when she started to bring me this data to show that NSP1 was in the nucleus, I thought it was a fail. I sent her back to the lab to try again. Something obviously had to be wrong. And she kept bringing this data back to me. And after the third time, maybe it was the fourth time, I don't know, I realized there was something really new and exciting here. And I tell you this because I want to illustrate Sam's perseverance throughout this whole process. She really went back to work and showed me and proved to me that her data was right. So when Sam started presenting this new data, she really quickly excelled at conveying these results to a wide variety of audiences. And she won the American Society for Microbiology Charles McCleskey Award and the LSU Health Top Speaker Award at Graduate Research Day. And she was also honored to give talks at 2 of the American Society for Virology meetings. Sam's work has been supported by the Ike Muslow Intramural Fellowship. And outside of the lab, Sam has taken several leadership roles as a representative on the Executive Student Council, and she also chaired the activity planning for Mental Health Awareness Week. Sam, throughout the years, has had ups and downs, but I think this seminar always allows our students to end on a high note. And moving forward, I hope Sam, as with all students, do the things that you think you cannot do. Fail at it, try again, do better the second time. It's only the people who never tumble are those who never mount the high wire. This is your moment. So Sam, go do great things with your PhD.
Thank you. And thank you for everyone joining me this morning. So what I'll do is I'll begin with a primer on rotavirus itself, as well as an introduction to the innate immune response before I move into our data that we've obtained over the last 5 years. So rotavirus is a non-envelope virus with a segmented double-stranded RNA genome, and it is the primary cause of severe diarrhea in young mammals. In fact, rotavirus can infect mammals of all species. Now, rotavirus infection typically leads to the generation of severe watery diarrhea and accompanied with a fever that can last up to 10 days. Treatment relies on preventing dehydration during that 10-day period. And of course, rotavirus is transmitted via the fecal-oral route. And these virions are actually very stable and hardy in the environment, and they can remain on hard, dry surfaces for up to 10 days. The reason being is this virus is a non-envelope virus with a triple-layered capsid structure. So these virus particles are very stable in terms of, and resistant to things like pH changes, temperature changes, and even some disinfectants. Rotavirus targets cells in the small intestine that are important for water and nutrient absorption, which is why many times people with rotavirus infection exhibit severe watery diarrhea. Now, of course, there is no treatment for rotavirus, but there are vaccines that have been introduced. Now, the vaccine efficacy does vary across the world from 85% to almost 100% efficacious. Now, the disease burden of rotavirus, however, occurs the highest in countries that are known as resource poor locations. Unfortunately, these are also places where the vaccines are not as effective for reasons not really well known. Rotavirus does cause over 200,000 deaths per year in humans, and it circulates in the human population as different strains. So rotavirus strains are constantly changing and evolving over time. Now, nearly every child will become infected with rotavirus by the age of 5. The adaptive immune response is very important for providing protection to rotavirus infection during the first and second infections. But the innate immune response is really what's crucial for controlling replication in children younger than the age of 5, because their adaptive immune responses are not fully developed. But before I can go into how rotavirus cuts off the innate immune response, I first want to talk about what your two immune responses are. So your immune system is comprised of two separate responses, the first being the innate immune response, and the second being the adaptive immune response. The innate immune response is sort of like an alarm system, and what it does is it primes your immune system to say, hey, we're infected, and this is the type of help we need. So viruses can actually trigger these alarms when they enter a host cell. And when these alarms are triggered, they warn other cells that there is an intruder, so these cells can start to take protective measures to prevent from being infected. They also recruit innate immune cells to the sites of infections so that virus can be recognized. And these innate immune cells then call in what I like to consider the super immune cells, or your adaptive immune cells. And adaptive immune cells are really important for ultimately stopping virus spread because they do things like kill the infected cell. So rotavirus can actually induce the innate immune response during infection. And so today we're focusing on what's called the interferon response, which is a major component of the innate immune response. The interferon response, as I just stated, like the innate immune response, is an alarm system. And the interferon response is what allows infected cells to control viral replication until immune cells can be recruited to help. Interferon beta specifically leads to the synthesis of interferon-stimulated genes. And ISGs are very important for inhibiting viral spread by doing things like cleaving viral nucleic acids, shutting down host translational machinery, and things like that. So how does rotavirus induce interferon? Well, during infection, the viral particle will enter a host cell and generate viral RNAs that can be recognized by pattern recognition receptors, or PRRs. PRR activation leads to activation of two separate signaling cascades that are both required for interferon induction. The first begins when IRF monomers are phosphorylated, causing them to dimerize and move to the nucleus. The second arm of the pathway begins when I-kappa B is phosphorylated. Now, I-kappa B typically sequesters NF-kappa B subunits in the cytoplasm, but upon phosphorylation, I-kappa B is recognized by beta-TRCP, ubiquitinated by proteins associated with beta-TRCP, and then proteasomally degraded. This allows NF-kappa B subunits to move into the nucleus together with IRFs to promote transcription of interferon beta. And just as rotavirus can induce this response, it's also able to inhibit the interferon response in order to promote viral replication. And it does so by encoding non-structural protein 1, or NSP1. NSP1 can work in one of two ways. It can either promote degradation of IRFs to inhibit their nuclear translocation directly, or it can promote degradation of beta-TRCP to inhibit NF-kappa B nuclear translocation. Now, what's interesting about NSP1 is that it is the least conserved rotavirus protein. And the reason why it is why it's not conserved is because of the C terminus. Now, the C terminus of NSP1 is what allows it to bind and degrade these two different substrates. And the NSP1 protein can actually bind or degrade IRFs or beta-TRCP, and this can vary in a strain-dependent manner. So before I talk about how we study how NSP1 inhibits the interferon response, I first want to introduce some of the commonly studied rotavirus strains, because these are the NSP1 proteins that I'll be talking about throughout the rest of the morning. So here, I'll begin with SA11-4F, which is a simian strain that has C-terminal sequences on its NSP1 protein that allow it to bind and degrade IRFs. Down here is an immunoblot from a paper published by our lab in 2016 in which cells were infected at an MOI of 5 with each of these rotavirus strains for 10 hours post-infection, and then whole cell lysates were harvested and analyzed by immunoblotting for IRF3, beta-TRCP, BP6 as a control for viral infection, and GAPDH as a loading control. So when cells were infected with SA11-4F, it was very clear that IRF3 levels are absent because IRF3 is degraded, whereas beta-TRCP levels were comparable to MOC because SA11-4F doesn't degrade beta-TRCP. However, there is a strain called OSU, which is a porcine strain that can bind and degrade beta-TRCP because of its C-terminal sequences. And so here I have OSU, WI61, and DS1, which all have very similar NSP1 proteins that degrade beta-TRCP. And when looking in their whole cell lysates of these infected cells, IRF3 levels were comparable to MOC, and beta-TRCP was absent, because again, beta-TRCP is degraded and NF-kappa B signaling is not intact. And finally, there are some NSP1 proteins that can degrade both. And one of these lab-adapted strains that has an NSP1 protein that's an excellent example of this is UK, which is a bovine strain. So when cells are infected with UK, IRF3 and beta-TRCP levels are both absent. And finally, no study would be complete without some sort of control, and that brings us to SA11-5S, which is another lab-adapted strain. It's also a simian strain derived from SA11, or similar to SA11-4F. And the thing about SA11-5S NSP1 is it lacks those C-terminal sequences that allow it to bind and degrade IRFs. So when cells are infected with SA11-5S, IRF3 and beta-TRCP levels are comparable to MOC. And of course, interferon induction does occur during infection with SA11-5S. And so that leads me to some of our work on trying to determine if NSP1 can inhibit IRF3 and NF-kappa B in a manner that doesn't involve degradation of IRFs and beta-TRCP. Now, some of that preliminary data stems from two studies, the first published in 2009, where they tried to determine the level of IRF3 inhibition compared to the level of IRF3 degradation. In this particular experiment, cells constitutively expressing ranitidine were co-transfected with plasmid encoding GFP-NSP1 and a PRD3 element that controls firefly luciferase. And these PRD3 elements are actually genetic elements where IRF3 transcription factors do bind to the promoter regions. Now, 36 hours post-transfection, poly-IC was used to stimulate these cells in order to enhance or induce IRF3 activation. Luciferase activity was then measured, followed by an immunoblot for phosphorylated IRF3, which is the active form of IRF3, and actin as a loading control. So here on the y-axis is the normalized PRD3 activity, so the level of IRF3 activation. And on the x-axis are the different proteins that were expressed in each of the cells. So I'll start looking at GFP alone, which should not inhibit or induce IRF3 activation. During poly-IC stimulation, there was an increase in PRD3 activity, indicating this IRF3 is active. But when cells expressed EW, UK, and RRV NSP1, which can all degrade IRFs, there was ablation of IRF3 promoter activation. However, when they looked at the immunoblots in the whole cell lysates, there was still a significant level of phosphorylated IRF3. So this indicates that NSP1 can completely abolish IRF3 promoter activation, but it doesn't completely degrade IRFs. How is that? Well, of course, with every study, we need to address the limitations because these limitations actually impact how we designed our own experiments. The first was they only examined NSP1s that promote degradation of IRFs. And the second is they did not assess the actual localization of phosphorylated IRF3. So it's very possible that this phospho-IRF3 present in these cells is still sequestered in the cytoplasm. However, that brings me to the other study published in 2015 that showed similar results. And so in this case, they're looking at beta-TRCP degradation and NF-kappa B activation. In this experiment, cells constitutively expressing ranitidine were co-transfected with plasmid encoding NSP1 and an NF-kappa B element that controls firefly luciferase. 36 hours post-transfection, cells were stimulated with TNF-alpha in order to induce NF-kappa B signaling, and then 4 hours after that, luciferase activity was measured. Now, in this experiment, they also compared the wild-type forms of NSP1 to the delta C13 truncated forms, which cannot bind beta-TRCP. The y-axis indicates the relative luciferase activity, so the amount of NF-kappa B activation, and the x-axis indicates which NSP1 was expressed in the cells. White bars represent the delta C expressing cells, and the black bars represent the wild-type expressing cells. And overall, when NSP1 degraded beta-TRCP, there was a significant decrease in the amount of NF-kappa B activation. However, they also assessed the level of beta-TRCP. And to do this, they co-transfected cells with plasmid encoding NSP1 and flag-tagged beta-TRCP. One of the reasons why they looked at exogenous beta-TRCP in these experiments is because of the fact that sometimes it's difficult to assess endogenous levels of substrate when just NSP1 is transfected into cells. And part of this could have to do with transfection efficiency, but at this time, the reasons for that is unclear. We just know that NSP1 during transfection degrades exogenous substrates much better. So 24 hours post-transfection, cells were immunoblotted for flag tag to detect levels of beta-TRCP, NSP1, and pCNA as a loading control. On the y-axis is the beta-TRCP level shown as a percent in the wild-type NSP1 expressing cells relative to the delta C terminally truncated expressing cells. And overall, it's very clear that NSP1 does not actually promote degradation, complete degradation of exogenous beta-TRCP. So this suggests that although NF-kappa B activation inhibition is complete, that beta-TRCP degradation is not. And so again, this study had some limitations, mostly that they examined NSP1s that promote degradation of beta-TRCP, with the exception of SA11-4F, but they also were assessing levels of exogenous flag-tagged beta-TRCP. As I mentioned, that can be a bit of a hindrance for some of the conclusions that you can draw, because sometimes exogenous proteins don't necessarily behave like their endogenous counterparts. But that leads me to our beginning of our work back in 2015, 2016. Cells were, initially we transfected cells with plasmid encoding tagged forms of NSP1. Then 48 hours post-transfection, I stimulated cells with TNF-alpha to induce NF-kappa B nuclear translocation. Then I harvested the cells, performed subcellular fractionation, and obtained a whole cell lysate, cytosolic fraction, and nuclear fraction. Then I immunoblotted for P65, which is an NF-kappa B subunit, NSP1 as a control, GAPDH as a control for the cytosolic fraction, and lamin C as a control for the nuclear fraction. And of course, our hypothesis was that in cells expressing SA11-4F NSP1, because it doesn't degrade beta-TRCP, we would see NF-kappa B, we would not see NF-kappa B accumulating in the nucleus. However, as with all things in science, that's not how things always turn out. So I'm actually abbreviating probably over a year's worth of work in this one figure, but I do want to just draw your attention to the fact that I've got WI61 and OSU here highlighted, which both degrade beta-TRCP. Now in this particular experiment, we were only able to see that P65 did not accumulate in the nucleus during TNF-alpha stimulation when cells expressed OSU or WI61, but none of the other tested NSP1 proteins. So based on that information, our hypothesis, we couldn't exactly prove our hypothesis, but we couldn't exactly disprove it either. Because one of the main questions we have to address here is, when you're doing subcellular fractionation, you're looking at an entire population of cells, rather than looking at the single cell level. So not every cell in these experiments were expressing OSU or WI61, or any of the other NSP1 proteins. So we wanted to know what happens during infection, because we wanted to use an immunofluorescence approach. Now, to do this, I infected cells for 8 hours, then immunostained for VP6 to identify which cells were infected, and NF-kappa B or IRF3, because in these experiments, I did look at IRF3. And again, our hypothesis is that SA11-4F infection will will lead to inhibition of NF-kappa B nuclear translocation, and OSU infection will lead to inhibition of IRF3 nuclear translocation. And our quantification here is that we determine the percent of infected cells with nuclear NF-kappa B or IRF3 from the infected cell population. So here are the results for the cells looking at NF-kappa B. On the left, I have the micrographs showing on the top row the untreated cells, and on the bottom row the cells treated with TNF-alpha. And on the right, I have a bar graph showing on the y-axis the percent of cells with nuclear NF-kappa B, and on the x-axis each of the experimental conditions. I'll begin with the mock-infected cells. In mock-infected cells that are untreated, NF-kappa B is diffuse throughout the cytoplasm, as we'd expect because this pathway is not stimulated. But upon stimulation with TNF-alpha, it's very clear that NF-kappa B then moves to the nucleus. And when we quantified this, nearly 100% of the cells stimulated with TNF-alpha had nuclear NF-kappa B. When cells were infected with SA11-4F, our experimental sample, we were able to see VP6 in many of the cells because this means that we have a high level of infection. But not every infected cell had nuclear NF-kappa B. In fact, only about 60% of the cells in the absence of TNF-alpha stimulation had nuclear NF-kappa B. And upon stimulation with TNF-alpha, that number did not significantly increase. So this suggests that there may be some blockage of NF-kappa B nuclear translocation even with infection with SA11-4F, which doesn't degrade beta-TRCP. So this does support our hypothesis. And in OSU-infected cells, it was very clear that in every cell that was infected, there was no nuclear NF-kappa B, even with the addition of TNF-alpha stimulation, indicating this blockage is complete. And we expect this result, right? Because OSU degrades beta-TRCP. Now, our results for IRF3 were a little different. On the top column here, I have just the IRF3 channels, shown in red, just for ease of viewing. And on the bottom row here, I have the merged images where IRF3 is in red and VP6 is in green. And overall, in the mock-infected cells, IRF3 can be detected diffuse throughout the cytoplasm and in the absence of stimulation. Upon infection with SA11-4F, we saw that infected cells actually did not have IRF3 in the cytoplasm at all. And when cells were infected with OSU, IRF3 could be detected in the nucleus of every cell that was infected. So overall, the results suggest that SA11-4F, although it doesn't degrade beta-TRCP, it moderately inhibits NF-kappa B nuclear translocation. But OSU, because it doesn't degrade IRF3, it does not block IRF3 nuclear translocation. And so that leads me to conclude part one, which is that SA11-4F can inhibit NF-kappa B in a manner that's independent of beta-TRCP degradation, but OSU cannot inhibit IRF3 nuclear translocation. And of course, with these experiments, we left a few stones unturned, including the activation status of this nuclear IRF3 and NF-kappa B. Because although they're present in the nucleus, it doesn't mean they're necessarily active. Now, part two of my talk, although it may seem like it's a little disjointed, the data for part two actually originated from data we found in part one. And so part two, we're going to address two questions. The first being, where does NSP1 localize in a cell? And the second being, what domains are required for nuclear localization of NSP1? Now, as Dr. Arnold mentioned during the introduction, rotavirus is a cytoplasmic replicating virus. Well, what does that mean? Well, once rotavirus attaches to a host cell, it enters an endosome where it then uncoats into the cytoplasm to become a transcriptionally active double-layered particle. These double-layered particles then generate viral RNAs, release them into the cytoplasm where they can be translated by host cell machinery into non-structural proteins like NSP1, or structural and non-structural proteins important for generation of viroplasms, which are the major sites of replication and assembly. And these viroplasms are all located in the cytoplasm. These newly assembled virions then egress through the endoplasmic reticulum and eventually leave the host cell. So because of its life cycle and the fact that no other studies have shown rotavirus proteins localizing to the nucleus, it's been assumed that NSP1 inhibits interferon induction from within the cytoplasm. Additionally, there were previous studies back in the 90s showing that SA11-NSP1 during transfection is diffuse throughout the cytoplasm and is diffuse throughout the cytoplasm and excluded from the nucleus. And in fact, these studies concluded that NSP1 is a cytoskeletal binding protein, which would help restrict its location to the cytoplasm. However, during our initial experiments looking at the localization of NF-kappa B, we found in our controls looking at NSP1 that NSP1 was in the nucleus. And so that led to a very long series of experiments trying to determine where NSP1 was, because we needed to know if there was an issue with our fractionation, if there was an issue with our controls. So initially, I wanted to compare expression of tagged NSP1 and confirm their subcellular localization. To do this, I transfected cells with plasmid encoding NSP1 for 48 hours, then harvested the cells, performed subcellular fractionation, and immunoblotted for halotag to detect expression of NSP1. GAPDH is a cytosolic control, and lamin AC is a nuclear control. So here I have the whole cell lysates, and these are the nuclear fractions. This row indicates which NSP1 protein is being expressed, and these blots indicate whether I was detecting NSP1, halotag, which is just a control because our NSP1 proteins are tagged, lamin AC, and GAPDH. And overall, in the nuclear fraction, we actually detected NSP1 from all of the tested rotavirus strains. And we did not detect GAPDH, which is very interesting. So this indicates our cytosolic fractions are clean of the cytoplasm. Now, with transfection, it can be a little tricky because you are overexpressing proteins in a cell. And unfortunately, sometimes this can lead to things like misfolding, mislocalization, aggregation. So we wanted to know what the subcellular localization of NSP1 was during infection, because sometimes having natural infection can kind of help us determine if what we're seeing during transfection is an artifact or not. So to determine the subcellular distribution during infection, I infected cells, then immunostained for NSP1 and lamin AC. Now, what we did differently to the other studies that looked at NSP1 using microscopes is we used a confocal microscope, which allows us to actually take a series of images called Z-stacks throughout the entirety of a cell. So here I have just a mock infected cell as an example. NSP1 will be in green in the next slide in the infected cells, and lamin AC is in red here. And we can see clearly that NSP1 or lamin AC forms a nice red ring around the nucleus, and anything within these boundaries of the nucleus can be assumed to be nuclear proteins. Now, when cells were infected with SA11-4F, NSP1, shown in green, was diffused throughout the cytoplasm and diffuse at low levels in the nucleus, which supports our data in the Western blot where we saw low levels of SA11-4F NSP1 in the nuclear fraction. And when cells were infected with OSU, NSP1 formed these very distinct foci throughout the nucleus, in addition to its cytoplasmic localization. And again, it seems like there's more NSP1 in the nucleus during OSU infection than SA11-4F. And this is also corroborated by our transfection data showing higher levels of OSU in the nuclear fraction. Now, one of the first questions we asked when we saw NSP1 localized in the nucleus was, how does it get there? Well, we know that IRF3 and beta-TRCP, which are both NSP1 substrates, we know they transiently localize to the nucleus. So we wanted to know if this nuclear localization was related to IRF3 or beta-TRCP binding and degradation. To do this, we used transfection model, because we wanted to compare the localization of full-length wild-type NSP1 proteins that can bind their substrates to the C-terminally truncated forms of NSP1 proteins that cannot bind these substrates. So starting with SA11-4F, during transfection we can see NSP1 in green, and it's localized diffuse throughout the cytoplasm and diffuse at low levels in the nucleus, supporting our data showing during infection even further. When cells expressed SA11-5S, NSP1's localization didn't change, indicating that IRF3 binding is not required for this nuclear localization. And when we performed the same experiment on OSU NSP1, we saw the foci form as we saw during infection. And again, when that C-terminus was truncated, where it cannot bind beta TRCP, these foci still formed. So IRF3 and beta TRCP are not the proteins that NSP1 is piggybacking on to gain access to the nucleus. But it seems that nuclear localization of NSP1 is conserved. So we wanted to know if the conserved ring domain of NSP1 may be involved in the nuclear localization. Now, before I jump into that data, I first want to introduce what ring domains are and what their functions are in a cell. Now, the NSP1 ring domain, we don't know much about it itself because we actually don't know the structure of NSP1, but we do know that in host ring domains, they're very important in what's called the ubiquitin proteasome system. Now, the ubiquitin proteasome system is a key player in regulating protein turnover in a cell. And the way it works is an E1, or ubiquitin activating enzyme, will pick up these ubiquitin molecules, which are basically molecular tags, and activate them so that they can then be transferred to an E2, or a ubiquitin conjugating enzyme. These E2s then bind to ring E3 ligases, which serve as scaffolds that bring E2 enzymes into close enough proximity to the substrate that's to be ubiquitinated. So these ubiquitin proteins can simply be linked on to the substrate into the form of a chain. Once these substrates are ubiquitinated, they're directed to the proteasome and degraded, or they can be trafficked throughout the cell. Now, we do know that the NSP1 ring domain likely plays a role in the ubiquitination of its substrates because NSP1 promotes degradation of beta TRCP and IRF3 in a proteasome-dependent manner. And so what does that mean for the way we design our experiments? Well, we wanted to start with these experiments using transfection with OSU NSP1 because OSU NSP1 forms those very clear nuclear foci. Forms these very clear nuclear foci. And essentially, we started out looking at 3 different mutations of NSP1. Again, as I stated, we don't actually know the full structure of NSP1, but we have an idea of what the ring domain looks like based on host ring domains. So we assume that NSP1 ring domain has this cross-brace motif that coordinates zinc ions to facilitate protein-protein interactions. It's supposed to serve as an interface. Now, what's important to these coordinated zinc ion, or these coordinated areas that bind these zinc ions, are these cysteine residues, because that is actually what maintains the integrity of this cross brace. So we initially started out looking at W41A, which is a mutation that would not disrupt the cross-brace motif and therefore should leave NSP1 as a functional protein that can degrade beta TRCP. But we also included C42A and C63A, which should disrupt this first cross-brace motif and render the ring domain non-functional. Now, we initially started out using subcellular fractionation. So here I have whole cell lysates and cytosolic fractions. The NSP1 protein that cells are expressing are indicated here. And here I have an immunoblot for NSP1, lamin AC, and GAPDH. So in the cytosolic fraction, NSP1 was detected very clearly. However, it's important to note that the level of these mutants were actually much higher than the wild type form. And the reason why it's important to note this is because when we look at the nuclear fraction, the level of C42A and C63A are significantly reduced compared to OSU, even though their total expression level is much higher. So this indicates that the nuclear localization of NSP1 is reduced when these C42A and C63A mutations are present. But the bigger question here is, do they still form foci? Well, OSU NSP1 is capable of forming foci in the nucleus, but surprisingly W41A was not. Although NSP1 was still diffuse in the nucleus, we expected to see the foci formation because that ring domain should be functional. So this is something that has yet to be explored. And with C42A and C63A mutations, NSP1 foci formation was lost, indicating that a functional ring domain is required for foci formation, but may not necessarily be required for nuclear localization. Now, to conclude part 2 of my talk, Overall, SA11-4F NSP1 is diffuse at low levels in the nucleus, and OSU NSP1 forms foci in the nucleus. This suggests that NSP1 could have novel functions in the nucleus that may or may not be related to interferon inhibition, and I'll actually touch on that in part 3. We also know that NSP1 nuclear localization does not depend on beta-TRCP or IRF3 binding, but that it may involve the ring domain. So that leads us to part 3. We know that NSP1 localizes to the nucleus. We think it has to do with the ring domain and not the C-terminus. But what are the functions of nuclear NSP1? Well, for part 3, we're going to answer 2 more questions. The first is, does NSP1 disrupt PML nuclear bodies during infection? And the second is, how does PML impact viral replication and spread? Now, I'm also summarizing a long series of experiments in which we looked for co-localization between NSP1 and different subnuclear structures. I'm focusing on PML because, as you can see in this table here, I have all the different subnuclear structures, at least the major ones, in a cell, along with the conditions in which we tested for co-localization, which included transfection and infection. And the only protein that we saw co-localization with was PML. Now, this top row here shows the micrographs where NSP1 is in green and PML is in red. And we have yellow arrows drawn through in order to generate what are called co-localization histograms. And the way these co-localization histograms work is the y-axis indicates the signal intensity of each of the proteins of interest, where the red line represents PML signal intensity and the green line represents NSP1 signal intensity. And the x-axis is the distance in which this arrow is drawn. So when looking at the mock infected cell, I drew an arrow through 2 specific PML bodies, and you can see each of their peaks here. When cells are infected with SA11-4F, there's no co-localization between NSP1 and PML. And although the graph kind of suggests that there is, I also confirmed these results by calculating the Manders overlap coefficient and the Pearson's correlation coefficient, and got values that were less than 0.4, showing that there is no co-localization. And when cells were infected with OSU, not only did we see clear co-localization in the micrograph itself, when we generated a co-localization histogram, the peaks of NSP1 and PML coincided with one another, indicating these 2 proteins are occupying the same space. Additionally, we saw a clear change in the morphology of PML bodies during infection with OSU. And so, before I jump into more of that data and how we explored it, I first want to introduce promyelocytic leukemia bodies, or PML bodies. These are distinct foci that have numerous functions in the nucleus that can range from tumor suppression to antiviral defense to serving as simply hubs where transcription factors congregate, or even as stress responses for sponging up ubiquitinated proteins in a nucleus. What we do know about PML, although we don't really know the mechanisms of these functions, we do know that PML is induced during stress responses like interferon signaling. PML proteins, upon interferon signaling, will begin to dimerize and recruit PML-associated proteins. And it's these PML-associated proteins that can allow PML bodies carry out their diverse functions. And in fact, the interactome of PML is well over 200 proteins. So PML is an interferon-stimulated gene. Interferon treatment will lead to an increase in the size and number of PML bodies. Based on this information, it's intuitive that viruses often antagonize PML bodies during infection. A great example of a virus that remodels PML bodies is adenovirus, and it uses E4-ORF3 to do so in order to disrupt interferon induction. So that led us to the question, does NSP1 disrupt PML body number and size during infection? To do this, we used immunofluorescence microscopy in infected cells that were immunostained for VP6 to mark which cells were infected and for PML. Now, in a typical cell, PML bodies are sphere-shaped foci, and there are 1 to 30 PML nuclear bodies per cell. So on the left here, I have the micrograph images showing PML in red and VP6 in green. And on the right here, I have two graphs. The first is the number of PML nuclear bodies per cell, and that's indicated on the y-axis. And the second is the area of the PML nuclear bodies, also indicated on the y-axis here. And overall, when comparing the infected cells to the mock-infected cells, during SA11-4F infection, we didn't observe a change in PML body shape, and we can clearly see that SA11-4F certainly infected the cell because of the amount of VP6 present. But what we did observe was a significant decrease in the number of PML nuclear bodies per cell. And although we didn't see a change in the area, that was to be expected because there was no change in shape. Now, when cells were infected with SA11-5S, which can induce the interferon response, we thought that there would be an increase in the size and number of PML bodies. In fact, this was supposed to serve as one of our controls. However, that is not what we observed at all. In fact, we saw no difference in the number or the size of PML nuclear bodies. Now, this could suggest that perhaps SA11-5S is also able to disrupt PML bodies. It just might not be as effective as SA11-4F counterpart. And when cells were infected with OSU, we didn't see a change in the number of PML nuclear bodies, which we kind of observed in the micrographs. But we did see a significant increase in PML body area, which is reflected by the change in shape because these bodies take on more elongated morphology. Now, as we've learned with NSP1, you can't assume that NSP1 from all rotavirus strains are going to behave in the same way because it's such a diverse protein. So we also wanted to look at PML body morphology and number in monoreassortant infected strains. Now, monoreassortant strains are very interesting to work with because they all have an SA11 background. So that is a strain that can degrade IRFs. However, they only differ in their NSP1 gene. So here I have a panel of reassortants that all degrade IRFs and a panel of reassortants that all degrade beta-TRCP. And essentially, I performed the same experiment in which these cells were infected and immunostained for PML, except this time it's in green, and VP6 in red. So here I have the SA11-4F-like NSP1 expressing monoreassortant strains on the left, and here on the right I have the number of PML nuclear bodies per cell shown on the y-axis of this dot plot. And overall, when cells were infected with an SA11-like strain, there was a significant decrease in PML nuclear body number, which is reflected by our previous results looking at just SA11-4F. However, when we looked at the OSU-like reassortant strains, we actually got a bit of an unexpected result. So the majority of them, we saw an increase in the PML body, we saw no change in PML body number, with the exception of just one. And what was interesting about this finding is that not only did we see a decrease in PML body number, we did not see a change in PML body morphology, indicating that not all OSU-like NSP1 encoding strains are going to alter the number of PML nuclear bodies per cell. And so what we had to do next was determine if NSP1 nuclear foci could form in the absence of PML. So we wanted to know, does NSP1 nuclear localization require PML? If it doesn't require IRF3 or beta-TRCP binding, and it may or may not require the ring domain, does it involve PML? To do this, we infected Hockat cells with the help of the SAP lab at LSU Health Shreveport. So we started out with a normal cell line, just the Hockat cells that express PML, and we transduced them with lentiviral vectors that either encode shNeg or shPML short hairpin RNAs. And so in the shNeg cells, we don't see a difference between the normal and these cells in terms of PML body shape, number, or size. But when cells are transduced with shPML, shRNA, we saw a significant decrease or significant knockdown of PML in these cells, and in fact an absence of PML bodies for the most part. We then infected each of these cell lines and immunostained for NSP1 and PML, or we performed a plaque assay to measure viral replication. And so here on the top column, I'm starting out with looking at nuclear foci formation. The top row shows the SH-neg infected cells, and the bottom row shows the SH-PML infected cells, where NSP1 is in green, PML is in red, and I have the merged images here. Now, I'm only focusing on OSU because of that foci formation is very clear phenotype, but we also perform these experiments in SA11-4F. And overall, in the SH-neg cells, OSU still form nuclear foci, PML bodies still changed in their morphology and their size, and of course there's still co-localization between the two. So our lentiviral transduction does not necessarily impact the way NSP1 behaves towards PML nuclear bodies. But when cells were infected, the SH-PML cells were infected, there was a loss of OSU NSP1 foci formation. And that was actually very interesting because we know two things. We know that PML bodies can sequester ubiquitinated proteins, and we think NSP1 ubiquitinates as substrates. So this becomes a question of, if you lose PML bodies in the nucleus, is that co-localization an area where NSP1 is perhaps ubiquitinating things at PML bodies? Or is it the other way around? Because there's two outcomes. The first, PML bodies can serve as sponges for NSP1 as a protective response for the host. The second is that NSP1 is promoting degradation of of proteins at PML bodies, and that's protective for the virus. However, we wanted to determine if rotavirus replication was impacted by the presence or absence of PML bodies. And again, as I stated before, we infected our SH-negs and SH-PML cells and measured viral titer, which is shown here on the y-axis in PFU per mil. And overall, there was no significant difference in the viral titer between SA11-4F and OSU infected cells in both of these cell lines. So this suggests that rotavirus replication is not impacted by the presence of PML nuclear bodies. But it's also important to know that rotavirus replication doesn't actually require a functional NSP1 protein in permissive cell lines. So cell lines that are very amenable to rotavirus infection don't necessarily have to have their interferon response shut down in order for infection to occur. And that's actually illustrated nicely from a paper published in 2005, in which cells were infected with SA11-4F or SA11-5S. And although replication still occurred, there is a very clear difference in plaque size, which is a good way to measure spread. Another good way to measure spread is by doing what's called a multi-step growth curve, where you measure viral titer over time during infection. So we did confirm that our PML knockdown is stable up to 3 days. In fact, it goes up to 4 days. And though I don't show those results here, more importantly, we measured the viral titer in OSU-infected SH-neg and SH-PML cells over the course of 72 hours, and we saw no significant difference in titer after multiple replication cycles. So this suggests that in permissive cell lines, rotavirus spread is also not impacted by the presence or absence of PML bodies. And so to conclude part 3, we know SA11-4F-like NSP1 proteins reduce PML body number. We know that OSU NSP1 disrupts PML body morphology. We know that PML nuclear bodies, their presence is required for NSP1 nuclear foci formation, but we do not see that knockdown of PML alters rotavirus replication or spread. However, again, that's in a permissive cell line. So if we were to use a less permissive cell line, would we see the same result? Additionally, what is the interferon beta induction level in these cells? And so, as I've kind of mentioned along the way while talking about our work over the last 5 years, we have many remaining questions that are actually very exciting. The first is, are nuclear NF-kappa B and IRF3 transcriptionally active in the nucleus when NSP1 is expressed? Just because they localize there doesn't mean that they're active. Additionally, we want to know, how does NSP1 gain access to the nucleus? When I performed a sequence alignment of NSP1, I actually didn't detect a canonical NLS. However, this doesn't mean that it can't get in there. The NLS in NSP1 could be non-canonical, or NSP1 could be piggybacking on a substrate that has a canonical NLS. Also, how does PML knockdown in less permissive cell lines impact viral spread? We saw in permissive cell lines that it does not. However, again, in permissive cell lines, the interferon response is not as important. And finally, is the proteasome required for NSP1 foci formation? We know that NSP1 promotes degradation of its substrates in a proteasome-dependent manner, and that it does involve the ring domain, but we don't know much else. Also, is the proteasome required for NSP1-mediated disruption of PML bodies? All of these questions would be very exciting to address in future directions. And so, with that, I would like to address our overall hypothesis at this time based on our data. We are proposing that NSP1 is capable of controlling the interferon response from two separate subcellular compartments. From within the cytoplasm, we know that NSP1 promotes degradation of beta-TRCP and IRF3 to inhibit IRF3 and NF-kappa B signaling. But within the nucleus, it's possible that NSP1 can sequester or disperse PML nuclear bodies in order to reduce the binding efficiency of transcription factors like NF-kappa B and IRF3 when they do reach the nucleus. And thus, this would reduce the rate of interferon beta induction. Also, what's more interesting about our data is that rotavirus so far is the only double-stranded RNA virus that's known to disrupt PML bodies. So we have uncovered an exciting aspect of PML body biology as well as virology. NSP1 is also the first rotavirus protein that has been shown to localize to the nucleus. Now, there is another non-structural protein of rotavirus that does antagonize a nuclear protein, so perhaps this is going to pave the way for some of those breakthroughs as well. We also know that NSP1 may have novel functions in the nucleus that are unrelated to interferon antagonism, and therefore we hope to reveal new functions of PML bodies during cytoplasmic viral infection. So with that, before I move on to the last part of the talk, which is always the sappy part, I do want to acknowledge my mentor, Dr. Arnold, fellow lab members, Dr. Kim, Kelly Brown, formerly Danielle, but she was great for the time she was with us. And then I'd like to acknowledge committee members, Dr. Harrison, Dr. Ivanov, Dr. Camille, Dr. Urachko, and Dr. Sherry for their never-ending support and contributions to this project. I'd like to acknowledge and thank the lab of Dr. Martin Sapp, as well as Dr. Martin Mudridge, for their assistance with the hot-cut cells and the subcellular fractionation. I'd like to acknowledge my funding sources, an Ike Muslow predoctoral fellowship that I received, that I earned back in 2017. I'd also like to acknowledge the IDEA and Board of Regents grants that Dr. Arnold received that has funded most of this work. And with that, I'll take any science questions before I move to the last part of the talk. All right, is anyone?
Sam.
Yes.
This is Kelly Tatchell.
Hi, Dr. Tatchell.
Very nice talk.
Thank you.
Great work. I have a couple of questions. One is, do you think the E2 is also targeted to the transported to the nucleus during this process, along with NSP1?
So,
or is that a possibility?
Of course, it absolutely is, because there are E2s present in the nucleus. There is an entire ubiquitin proteasome system in the nucleus. I mean, this is not just a cytoplasmic process. So it's possible that NSP1, which is what points at that W41A mutation, it's not clear that that's exactly what it's for, but it's possible that it is for binding to an E2. So when we mutate it, of course, we see loss of foci formation.
And the other question is NSP1 ubiquitinated? Because that would explain a lot if that were the case.
Yes. So we, yes, we do. We know that that NSP1 is post-translationally modified to be ubiquitinated, but the only part about that we don't know for sure is, does NSP1 directly ubiquitinate its substrates? That's all. But yes, NSP1 does get ubiquitinated, and that would explain some of that aggregate formation we see in the nucleus. And it could also explain why we don't see aggregate formation for some NSP1 proteins in the nucleus, because, again, ubiquitin is important for protein turnover, and sometimes the rate of turnover can vary between different proteins. But, of course, that type of idea needs to be explored in the future.
Yeah, it's hard, too, to find the right lysine. It can be a fool's errand sometimes chasing those around.
Yes.
Thank you.
You're welcome. All right, I don't see any more questions in the comments, so I'm going to move on to the very last part of this talk, and probably the sappiest part, but that's all right. So one of the things I've learned in my journey as a scientist is I love bench work, but I also I just feel joy and happiness when I'm doing science. However, as Dr. Arnold mentioned, I've had to overcome quite a bit, and sometimes life gets in the way. So one quote that's kind of carried me through my journey is, "Strength grows in the moments when you think you can't go on, but you keep going anyway." Because science really is a journey. And so I'm from Shreveport, Bossier. I graduated high school here. Heck, I went to elementary school here at little old R.V. Kerr Elementary in Bossier City. And I really am a Shreveporter to the core. I mean, we are friendly folk. We do love our outdoors. And, and of course, a lot of my life choices, at least the beginning before my science journey started, followed the same trajectory of most of the people that I grew up with. Now, a PhD really is a marathon. I don't think people really appreciate how long these things take, but here's a picture of my kids in 2015, right before I started my PhD program. And these are my kids now in 2020. They grew up completely in the last 5 years.
Hello.
And fortunately with my kids, they've been a huge source of my strength. And actually, as many of you probably already know, the first child I had was back in 2007 when I was 17 years old. And I started college in 2008 and had my other child in 2010 shortly after, before I was 20. Now, based on that information, my kids have only known a mother who was in college, and I've only known how to go to college with kids. And most people ask me, how do you do it? Well, I'm telling you now, I've spent many a day prepping for exams while holding a baby. And the way I explain how do you do it is with a freaking good sense of humor, and of course, a couple of really good kids. So, and I think one of the hardest parts of this last part of my PhD program has been that our family dynamics are changing a bit. But I do want to acknowledge my my former partner, for his role in my decision to even go to college and to stay in college. And I'm also grateful that he also went to college. So I think the two of us set a really good example for our kids to just say, hey,
Stop.
I can't.
Why are you crying?
It's just great. To just say, hey, yeah, life is tough and maybe you start from the bottom, but nothing's impossible. And of course, no dedication would be complete without mentioning my family, which I'll start with my parents, Mary Ann E. and Eric. And of course, here's my dad doing what he does best, teaching kids about rockets, because that's what he did in the military. And it's that love of his work that actually instilled my love of science.
Is that Grandpa?
Now, here's my dad, spreading that love of science to his grandkids. As you can see, they're all very much into what he's showing them. And he's just showing them some of the rocket launches he used to work at when he was in the military. But here's also my mother. And though she may be tiny, she is fierce. And That's where I get that from. All that fierceness, all that strength, all that dedication. I get that from my mother. This is my Uncle Brett. He is my dad's identical twin brother. And I don't know if you can see his shirt, but it says, "Who are these kids and why do they keep calling me Grandpa?" And that's definitely because my kids confuse him for Grandpa all the time. But I'd also like to acknowledge my Uncle John, along with my other aunts and uncles, because we actually lost my Uncle John tragically in 2016. And so that was a very hard time for the whole family. Now, I also have a sister, a little sister. This is my favorite picture of us. It's the best. And of course, we've gone through everything together. Babies, weddings, everything. So here's us in 2007 when I was having my daughter Roxanne, and of course she's happy. And this is us in 2016 when it was her turn to have a baby. And this actually was the night before my Methods 2 exam. I rode to Dallas and stayed up all night studying in the car and drove straight home. I literally got to hold my nephew and put him down and go take my exam. And of course, she's a lot happier here because I'm sure they gave her better drugs. But here's her boyfriend, Taylor, who has become like a little brother to me. And this is my tater tot, my nephew, Carter Mack. But I don't just have that family. I have a whole other half of the family, too, that we call the Murphy clan, because we're a whole gang of people. And although our family situation might be changing a little bit, they'll always be my family. So Poppy, Sheila, Mark, they're the parents. My sisters, Heather, Kristen, Chesley. Of course, there's my sister by blood. And of course, all of my nieces and nephews, Dylan, Riley, Hunter, Lily, Brooklyn, and Caitlin. I would like to just say that they all, I know I'm the first doctor in the family, but I hope I'm not the last. And not only do I have an amazing family by blood and by law, I have family by choice. So I want to start with my sister by choice, Ariel. We grew up together. We met when I was 11 years old, and we've stuck together ever since. I can't think of anyone better to travel with and to get trapped in hammocks with. And then there's also my die or die, Becky. I love her so very much. We met in middle school as well. And Becky, Jessica, and I have sort of formed our own little mom squad. We have hats. You should join us. But behind every strong, empowered woman is a girl gang of other empowered women. And so this is my family. These are the loves of my life. I adore them all. They're all so amazing and so supportive. Megan, Erica, Lucille, JK, Kelly, Christina, Janae, Haley, Francine, Alicia, Jess, Lauren, Sarah, and Vanessa. And of course, Marshall's just kind of the honorary member of our girl gang. But my journey as a scientist would never have been possible without the people at the Health Sciences Center. And I'll start with my mentor, Dr. Arnold. She is a force to be reckoned with. If there's anyone you want to learn something accurately and correctly from, it's her. Because what she does, what's so great about her as a mentor, is she dedicates herself to her work. She really throws herself into it and teaches you how to dedicate yourself too. And she provides a great example of a strong woman in science, of a woman who doesn't know, who doesn't back down, who won't quit, who doesn't give up. She's also hilarious and great to be around. And of course, because of Dr. Arnold, I think that she's created other great scientists like Kelly and I. Kelly is my lab mate, my partner in crime on a Monday through Friday, 9 to, or more like 8 to 6 basis. And so we have a lot of quotes from our wonderful mentor that we use all the time to stop ourselves and make us think. One of our favorites as well, "You're not wrong." But I also was a scientist before my PhD program, and This is difficult because we lost her very recently. Dr. Hart was my master's mentor, and actually one of Dr. Tatchel's students. Small world, right? She was just this light, this love. Everything about her was just warm and caring. And I really wish she could be here today. But I had a whole family at LSUS. So these are Dr. Hart's kids. Amber, Chris, and Liz and I were all together in the Hart Lab. Madison was an honorary Hart Lab member, and I'm so happy to see him graduating soon. And of course, Peyton, always great for hugs whenever I run into him on campus, and my study buddy and other partner in crime and weird adventures. And my LSU Health family, because that's what y'all are. You're a family. And there's so many of you. I can't even name all of you because there's so many of you wonderful people. But all the beers with buddies, all the get-togethers when we were out at conferences, birthday parties, Halloween parties, just getting together to kind of show in solidarity for scientists in the southern U.S. as well as women in STEM. You're all amazing, whether you were a student, a postdoc, a staff member, or another PI. You've all been an amazing support system. And so with that, I'd like to close and just say, I love everything about being a scientist, and it's because of people like you and work like this. Thank you.
Sam, in behalf of the department, I would really like to congratulate you to your achievements. This was a great talk. I hope you will be able to continue your journey in science and contribute to our knowledge that we are trying to expand over the years. All the best for you in the future.
Yes, thank you so much.
Congratulations, Sam.
Thank you.
Congratulations. Great job.
Thanks.
You did it!
Yeah! We can scream more this weekend.
Definitely.
Yay!
Okay. All right, I'm going to log off now. Thank y'all so much for coming. Bye.
Bye.